Well, I would like to welcome everyone to Brain Research Foundation's lunchtime lecture. I'm Dr. Terry Constantine, and I'm the CEO of BRF. I'm very pleased to be introducing Dr. Lindsay DeBaise. Dr. DeBaise is an associate professor at the University of California, Los Angeles, and a 2021 BRF Seed Grant recipient. Today, she will be talking about how her lab is studying brain cells called microglia, and she is uncovering how important they are in overall brain health. Understanding this may reveal new strategies for protecting the brain as it ages. I'm sure you will enjoy her talk, and there will be time at the end for questions. So any questions you might have, please type them into the Q&A section at the bottom, and we will try to get to as many as possible. Now, let me turn it over to Dr. DeBaise.
Thank you, Terry. Good morning or good afternoon, everyone, depending on where you're joining from. I am really excited to have this opportunity to tell everyone about some of the research that's going on in my lab, and also to highlight what a key role the support from the Brain Research Foundation played in helping us build momentum in multiple research directions. So I'm going to start by talking about some different cell types that are present in the brain. Many folks may be familiar with neurons. These are the electrically excitable cells within the brain. And neurons are connected to one another through these special connections called synapses. So one neuron might be receiving thousands of synapses from other neurons. And this is really where we think information is stored within the brain. So new synapses can be created, synapses can be removed depending on what you're learning and what experiences you're having. And that's the way that we will store memories and store new information in the brain. These cells are also electrically excitable, and so they can be wired together in these complex electrical circuits that let information flow throughout the brain. And so this is one of the reasons that you'll often hear people make the comparison between your brain and a computer. And there are these important similarities that both your brain and computers have a way for storing information and a way for wiring all the different pieces together in these highly complex circuits. But what I'm going to tell you about today is that there are also many ways in which your brain differs from a computer. And one of the key ways that your brain is really different from a computer is these elements that store information, the neurons, they're really only half of what's present in your brain. So neurons make up about 50% of the cells in the human brain. The other 50% of the cells in your brain are these non-neuronal cells that we refer to as glial cells. And we're going to talk a lot about those today. So glial cells, I do want to just give a quick historical perspective. Glial cells were discovered a long time ago. This is actually a hand drawing from the late 1800s by a very famous neuroanatomist, Ramon y Cajal. And here he's drawn some pictures of glial cells that are interacting with neurons. And he was very intrigued by these glial cells and what they might be doing for brain function. But ultimately, what happened in the course of neuroscience research over the next 100 or so years was that most of the attention was paid to the neurons, and the glial cells were largely kind of forgotten about. They were assumed to mainly be there to kind of hold the neurons together to provide structural support. And in fact, the word glia comes from a Greek word for glue. So for a long time, the idea was that these glial cells are just there to kind of hold everything together. I hope by the end of my presentation that you will be convinced that glial cells are doing more than just holding the brain together, and that they actually are very important in determining brain health in many contexts. So what I'd like to tell you about today are the following things. I'm going to tell you a bit more about one type of glial cell, microglia, These are the main glial cells that we study in my lab. So I'll give you a little introduction to these specific glial cells. And then I'd like to tell you about two lines of research that we have going on in the lab that I'm particularly excited about. One is looking at how microglia interact with neurons during brain development. And then the second is looking at how microglia change during aging and what that might mean for whether or not we start to have decline in cognitive function. So let's start with introduction to microglia. For a long time, we thought that microglia were mainly carrying out immune functions within the brain. You'll often hear people call them the immune cells of the brain. And it is true that that is an important part of what they do in the brain. So if any bacteria or viruses get into your brain, these would be the cells that are going to find them and get rid of them. And here I'm just showing you a visual example of this. So these were some studies that were carried out actually using zebrafish. And the reason is that zebrafish are transparent for kind of a large portion of their lifespan. And so that allows you to do some live imaging of cells within the zebrafish brain. And so what you're seeing here are some E. coli bacteria that have been labeled in red, and then the microglia in green. And they've done some imaging over time. And what you can see here is that the microglia are really seeking out those red E. coli bacteria and actually engulfing them. We call this process phagocytosis, but they're eating these bacteria to remove them from the brain. So this is just to say, yes, microglia do have important roles as immune cells in the brain. But in the last 15 or so years, we've learned that they are also doing a lot of additional things besides just making sure that we don't have any viruses or bacteria present. So what I'll tell you about today is we've learned a lot. We're beginning to learn about how microglia actually interact with neurons and affect the ability of the neurons to store information or send information throughout the brain. So for example, microglia can influence formation of new synapses or removal of synapses. So that's going to impact information storage. And they can also influence the electrical excitability of neurons. So that's going to affect transmission of information throughout the brain. So I think we can make a strong argument that if we really want to try to understand learning and memory and behavior, and also situations where those things start to break down, we do need to understand what role microglia are playing. How are microglia neuron interactions influencing these processes? These interactions between microglia and neurons are important for every population of neurons in the brain. And that's because microglia are present everywhere in the brain. This is just a visual to illustrate this. This is what you're looking at here is a cross section of a mouse brain. And all the microglia have been fluorescently labeled. So each little white dot that you see here is a microglial cell. And so what I hope you can appreciate is that there are microglia just evenly tiled all throughout the brain. So no matter what population of neurons we're thinking of, microglia are going to be there, potentially interacting with those neurons. An early discovery that my lab contributed to that really has shaped what research we've done moving forward is the discovery that even though microglia are present all over the brain, they're not totally equal all over the brain. So there are some intriguing differences in microglia in different brain regions. And that suggests that they may be having kind of unique or special interactions with neurons in specific brain regions, depending on which brain region you're looking at. In my lab, we have primarily been looking at microglia and how they interact with neurons in some circuits that are called the reward circuits. These are circuits that involve a number of brain regions that are deep in the brain. And I'm going to be telling you more about microglia in two different parts of these circuits. One is a brain region called the nucleus accumbens. And in this brain region, microglia tend to be very abundant, and they also tend to have a lot of branching processes. Where I'm going to tell you about some work that we're doing looking at how microglia in this brain region influence development of neurons in that brain region. And then we'll also share some work that we've been doing looking at how microglia in this brain region called the ventral tegmental area, how they change during aging and what that might mean for neurons that are nearby. Microglia in this brain region are less abundant. There are fewer of them. They don't have as many branching processes. And so, again, they may have some distinct ways that they interact with neurons and influence their health. Okay, so let's start with thinking about how microglia might be affecting neurons during development within these reward circuits. So we had reason from other experiments that we were doing in the lab. We had some reason to suspect that maybe microglia were helping neurons in this brain region, the nucleus accumbens, to mature. And a way that we tried to look at that further was by analyzing genetically modified mice that lack microglia. This allowed us to look at things like how are the neurons different, or how are the synapses different if microglia just aren't present. What we did to look at the synapses is we used a technique called immunostaining. It basically lets you label different structures in the brain with fluorescent proteins so you can look at them and see how many there are, where they are. So we used one label to one labeling to look at what we call the presynaptic terminal, and a different labeling to look at the postsynaptic terminal. And together, these things can show you a connection between two neurons. When we looked at the nucleus accumbens of mice that have microglia, you can see all these green and red dots. And then compare that to the nucleus accumbens of mice that don't have microglia. It was looking like there were a lot fewer of these red and green dots, these synaptic structures in the mice that lack microglia. And in fact, if we went in and we really carefully tried to count and just like reconstruct each of these synaptic structures, we did find that in the mice that lack microglia, there are fewer synaptic connections than the mice that have microglia. And this was at a time period in development in mice that we refer to as postnatal day 20 to 22. This is about the equivalent of like a 12-year-old human. So, they're not babies, but this is still a period when the system is maturing and fully coming to its adult-like function. So this was really surprising to us. Wow, if microglia are not there, it seems like synapses are not forming as well. We wanted to look at this using an additional technique, and we used a special technique called electrophysiology. This actually lets you record electrical currents from neurons. And so if we record, what you're looking at here are some recordings of electrical currents in neurons from this brain region, either at that postnatal day 22, that about equivalent to a 12-year-old human or a little bit younger. And each of these downward lines is an event where a signal has been transmitted at a synapse. So in a normal mouse, that has its microglia, what you can see is that, okay, at this stage of development, there are some synapses, and then there's even more synapses as the mouse is getting older. So this is a period of generation of new formation of new synapses. But again, in these mice that don't have microglia, we were just seeing a lot fewer of these synapses. So these two different ways that we looked at synapses, we were finding that if microglia aren't there, it seems like not as many synapses are forming. Does that matter at all for behavior? Does this have any consequences if fewer synapses are formed? We think it does have important consequences. So we've done a number of behavioral tests on mice that were lacking microglia. And I'm just going to tell you about one of those tests that tries to look at whether the mice are experiencing more anxiety. So the way this works is a mouse is placed in a box or an arena, and we have a video camera up above that lets us see where the mouse is moving. And these are examples of the path that a mouse has taken over this recording period. And what you can see is that in a mouse that has microglia, it explores all over in this box. It kind of checks out all the corners, runs across the middle, runs around the edges. But that's kind of different than what we see in a mouse that didn't have any microglia. It's really, really running mainly around the edge of this arena and not going into the center. This is usually this is a key indicator that a mouse is in a higher state of anxiety if it's not wanting to go out in the center of this arena and really prefers to just stay on the edges. So here, this is just a quantification over here that these mice that lack microglia were spending a lot less time in the center. So we think that there's an important consequence of the microglia not having their not being present and not helping with synapse formation, that we end up with a situation where mice are more anxious in adulthood. This was very intriguing to us, but it is true that it's a bit of an artificial situation to just completely lack microglia. So what I told you is that, if microglia are present, great. But if they're not present, we have a situation where there are fewer synapses and more anxiety. This suggests that microglia are doing something important, but it's not necessarily something that we would see happening in humans. There aren't really situations where humans are just going to completely lack their microglia. But there are a lot of situations where, unfortunately, humans will face challenges early in life that might alter the microglia or interfere with microglial function. And an important example of that is early life stress. So the future directions that we're taking now are trying to understand, okay, what if microglia aren't necessarily absent, but what if they've been perturbed by some substantial challenge early in life? Are there going to be similar consequences for the ability of neurons and these circuits to mature? This is work that's ongoing, but I'll tell you some of what we're finding so far that I think has really important implications for mental health. What we're doing to follow up on this is we're using a model of early life stress. In this model, what happens is there are mice that are growing up in a normal environment where there's plenty of materials for the mother mouse to create a nice nest for the young to be raised in. And then there are animals instead where there are a lot fewer resources to create a good nest for the animals. And this is meant to model the adversity and scarcity of resources that individuals sometimes face early in life. This difference in the environment is present from when the mice are 4 days old to when they're 11 days old, and then everything goes back to normal. So it's just this restricted period during the mouse's development. But that's enough to result in heightened anxiety in these mice once they grow up. So this is again that test where mice just go in the box and we see if they stay near the edge or they go near the center. And mice that grew up in this more restricted environment do show elevated anxiety in adulthood. So that's sort of similar to what we were seeing when microglia were absent. So we'd like to know, does this difference in presence of stress or lack of stress when you're growing up, does that affect the microglia in the brain? The way that we've been looking at this is we have some techniques that let us purify the microglia from this brain region that we're interested in, the nucleus accumbens. The technique that we're using is something called flow cytometry, and it basically just lets you take your cells of interest from a mix of tissue and purify them. We did this from mice from brain tissue that was collected during the stress period, but also collected much later in adulthood. And what we wanted to do was ask, are there specific genes in the microglia that are either turned on by the stress or turned off by the stress? And we can look at this using a different technique called RNA sequencing. This is data that we're still actively analyzing. So these are just kind of early findings. But what we are finding is that there are a lot of genes in microglia that are either turned on or turned off by this early life stress. So some examples are, there are a lot of genes that are very important for kind of the overall microglial identity that appear to be turned on to a different degree in the mice that have experienced stress. There are also some genes that are involved in microglia maybe removing, eating synapses that are turned on by this stress. And then these are some genes involved in microglia sensing how active the neurons are. And a lot of those were really turned on from these mice where we analyzed the microglia during that stress period. Instead, out at this adult period, quite a lot after the stress, we were seeing a lot of genes in microglia that are involved in keeping those synaptic connections stable that were turned off by the stress. So I know this is a lot of different gene names, but the most important message here is that there seem to be a lot of strong impacts of stress on which genes are turned on or off in microglia. So the take-home messages that I'm hoping you all get from this part are that these microglia in this particular region called the nucleus accumbens, we think they're supporting synapse formation during development. If the microglia are gone, there are some deficits in this synapse formation, and that seems to be linked to anxiety. And also that early life stress has a really profound impact on microglia and is very likely influencing how they will support neuron synapses and neuron health. So our goal moving forward from this is to understand if some of these changes that we're seeing in the microglia might be the driver of why individuals who've experienced early life stress are at increased risk for anxiety and other mental health issues that include substance use disorders later in life. If we can understand this, that might provide opportunities to intervene in some way and help reduce risk for these mental health challenges in individuals who've experienced severe early life stress. So this second part is going to be shorter. But I'd like to take us to the other end of the lifespan, and talk about some things that we're finding in terms of how microglia change during aging and what that might mean for whether you have cognitive preservation or whether you start to have cognitive decline and challenges with memory, et cetera. So I'm going to tell you about two things. First, I guess I should just say microglia change a lot during aging. And I want to tell you about two things that we've observed that really caught our attention. Here we're looking at microglia in that ventral tegmental area. This is actually where your dopamine neurons are located. And one thing we noticed that was happening to microglia in this brain region during aging is that they're filling up with these aggregates of proteins and lipids. So here you're seeing a microglial cell in green, and then this aggregate is here in red, and here you can just see the aggregate by itself. This was like really noticeable. We were like, oh my goodness, look at the microglia in this brain region are really filling up with these aggregates. And it seemed like that was maybe causing the microglia to proliferate and divide and increase in number. So here you can see a couple examples of microglia that are dividing, and then kind of what happens to the aggregate. Most of the time, it's just like one cell ends up with the aggregate and the other cell doesn't have aggregate anymore. So we think they might be, part of this might be that the cells are trying to dilute out these aggregates. So this is something that happens to microglia during aging that caught our attention. And one additional thing that really caught our attention was that it seemed like microglia during aging were beginning to contact nearby neurons. So here I'm showing you in particular the dopamine neurons that are present in this brain region in red. And you can again see the microglia in green. And these are just a few examples of the microglia of interactions that we see where it seems like the microglia are physically contacting the dopamine neurons. What we were intrigued by was that it seems like this contact is increasing during aging. So we were seeing a lot of this contact increase between a mouse tissue from mice that were 6 months of age to 12 months of age, and then a bit further by 18 months of age. This would be kind of equivalent to about a 30-year-old human, 45-year-old human, 65-year-old human. So we're not talking about way out at the end of the lifespan. This is stuff that's starting during middle age. And it caught our attention that it seemed like this contact of the dopamine neurons was happening more in the ventral tegmental area compared to this neighboring region called the SNC. They caught our attention because there are dopamine neurons in both of these regions, but the ones in the ventral tegmental area seem to be more resilient during aging. So we thought maybe this is a protective interaction. Maybe the microglia are sensing that the neurons are under a bit of stress during the aging process, and they're contacting them to help support them in some way. Okay, so those are two things that we saw about how microglia change during aging. They fill up with aggregates and proliferate, and they start contacting the dopamine neurons. We are nerds, and so we think those things are exciting no matter what. But in general, to convince other people that these findings are interesting and to see whether this might allow us to improve brain health during aging, we needed to try to understand if these microglial changes were linked to brain function in any way. And we think they are. The way that we studied this is by looking at reward memory and cognitive flexibility in aging mice, and then seeing whether the reward memory and cognitive flexibility in aging mice is connected to these microglial changes that we were seeing. So I'll tell you briefly about the way that we tested reward memory and cognitive flexibility. In this behavior test, we place a mouse in a very large arena, And there's a little bowl where mice can dig for a food treat, a little food reward that they really like to eat. So the mice go into the arena, they find this little bowl where they can dig for a food reward. Then we take them out of the arena and we wait for either 2 hours or 24 hours and then put them back into the arena. And that same bowl that has the food treats is present, but there are also three additional bowls that are kind of distractor bowls. They don't have any food reward in them. And we can see where the mouse goes and determine whether or not the mouse remembers which bowl has the treats. We do this test every day, and every day the specific location where there's going to be a bowl that has treats changes. So the mouse has to have both good memory, but also enough cognitive flexibility that they can forget where the treat was yesterday and remember where's the treat going to be today. When we look at how mice behave in this test, 18-month-old mice, so late middle-aged mice, do not perform as well as adults, particularly if we ask them to do this after a longer delay. And now we can go and see, is there any relationship between the 18-month-old mice that are doing the best and the 18-month-old mice that are not doing as well, and what's happening with their microglia? So first, let's think back about, we saw that when microglia are accumulating these aggregates, they start to proliferate, so there are more of them. How does that relate to a mouse's behavior? What we ended up finding is that here, if you focus on these white triangles, these are the mice that are late middle-aged mice. The mice that had more microglia in their VTA were doing worse on this behavior task. So it seems like whatever is driving this process in the microglia, it's associated with declining cognitive function. What about that other thing that we noticed where we saw microglia were contacting dopamine neurons? Here we actually find the opposite, where the 18-month-old mice that had more physical contact between the microglia and the dopamine neurons were doing better on this behavioral task. So this really indicates that microglia, what's happening with microglia is pretty tightly linked to what's happening with your reward-based memory and cognitive flexibility, and that there might be multiple ways that we could try to a little bit change the way that microglia are behaving during aging to try to improve cognition. And so this is, I'll just kind of re-summarize that. What I told you about microglia in this brain region during aging is that they're filling up with these aggregates and proliferating. That seems to be associated with worse cognition. But they're also contacting these vulnerable dopamine neurons, and that was associated with better cognition. Overall, where we want to move forward in this line of research is trying to understand what is making these different changes in microglia happen during aging. What is it that's causing them to fill up with aggregates? Is that something that we could try to decrease? And then what's causing them to reach out and contact these dopamine neurons? Is that something we could boost because it seems maybe like it's protective? And ultimately, understanding these things might help us design interventions where we could help preserve cognition. And we also think that this may be important for decreasing your risk for neurodegeneration during aging, particularly Parkinson's disease. So I hope I didn't run through that too quickly, or that it was fairly digestible. I really love answering questions, so I'm happy to answer any and all questions that you might have. What I'm showing you here are a couple different pictures of the students and postdoctoral fellows who have been present in the lab. And I'd really like to highlight that for my lab, we received the seed grant from the Brain Research Foundation when my lab was about 2.5 years into its development. And that can be a really, really critical juncture for a lab. You have some ideas about a new area that could be really important to explore, but you really need a vote of confidence and some financial support to try to begin exploring those ideas and get a foothold. And for us, the seed grant absolutely had that function. It allowed us to carry out some key initial experiments in both of those lines of research that I was telling you about. And that then ultimately helps you go after and try to secure additional funding to keep following those lines of research up. And so, this certainly helped us in our journey to secure research funding from NIH and other disease foundations. So it can be a really great catalyst for labs that are trying to explore a new direction, and also labs that are kind of in that early stage of building up their research program. So we were very grateful for that support. I'm going to advance us to this final slide for Q&A. And I'm really happy to take any questions that anyone may have about microglia in general, the specifics of the research, wherever we want to go.
Great. Thank you. First, let me say thank you so much. That was really interesting. I'm going to be looking in the Q&A section and asking you some questions.
Great.
One question is your lab also looking at the potential benefit of the Exelon patch? Maybe you could, for those that perhaps are not familiar with that, could you first explain it and then answer the question?
So I don't know if I'm familiar with the Exelon patch.
Okay. Uh,
So I guess the answer to the question is that at the moment, we
no.
haven't looked at the benefits of that. But what I would say is that um, microglia as cells are really sensitive to many different things that can be happening with the body, right? So that can include uh, diet, exposure to pollution, sleep, your sleep quality. And so certainly I'm a little bit guessing here that the patch may be something like either a pharmaceutical or like an herbal something that can be associated with improving health that you might wear on your skin somewhere. But certainly things that are coming from the rest of the body or that you're doing to try to improve the health of the rest of the body are also very likely to influence how microglia are behaving and how microglia are interacting with neurons. So I apologize that I'm not familiar with the patch, but it certainly is very possible that um, anything that you're doing to improve health of the rest of your body would influence what's going on with microglia.
And then there's a question from kind of in the beginning of your talk, or more in the beginning, the early life stressors. Is
Mhm.
there an amount of decrease of the microglia until you see the anxiety effects?
Yeah, so I guess the question would be like, would you need to have all of your microglia gone to see this effect, or would just eliminating half of the microglia be sufficient to induce some anxiety like behaviors? I don't know the answer to the question because we don't really have a great way to um, control in a precise fashion the number of microglia. At the moment, we have tools or we have techniques that can let us either have the microglia present or have the microglia absent. But I think in the context of the early life stress, we aren't necessarily seeing any changes in microglial number. So, like the number of the microglia is staying the same, but the stress is really affecting which genes are turned on or off in the microglia. So even if we don't change their number, if the stress has interfered with their ability to interact with the neurons and maybe help them mature, that may have really important consequences for how the circuits develop and whether an individual is likely to experience anxiety or other mental health challenges. Hopefully
And
that
kind of a
answers the question.
follow-up on that, because you mentioned the genes turning on or off. Is there anything that, during the stressor, is there any link the genes with any neurological disorders, the ones that are turning on and off?
Um, yeah, I mean, this that data set where we're looking at the genes that turn on and off is still work in progress, but I assume we are asking about, there are certain genes that genetic studies have linked to, various risk for neurodevelopmental disorders or some risk for other mental health challenges. Um, I don't know if there are specific genes showing up in our data set that have been really big hits in these human genetic studies, but that is absolutely something that will be really important to look at.
And what about sex? Does that determine how or influence changes?
Yeah, this is a great question. We include male and female mice in our studies. In the early life stress, it does seem to be the case that we're seeing some sex differences, that maybe some of the effects could be a bit more profound in the males than the females. But again, this is still like it's work in progress. So, um, take everything with a grain of salt. But I think that is that is a super important question for the microglial field and neuroscience in general, is being careful to understand the things that we're studying, are they happening the same way in males and females? It's definitely also a very relevant question for our studies of what microglia are doing during aging. I mean, there are differences, there are sex differences in how high your risk is for different neurodegenerative diseases, for example. Um, and so that's also a place where we're trying to be very careful to analyze and see whether there are any sex differences in what we're observing.
And then there's a question about microglia and does it have an effect on oligodendrocytes or myelin?
Yeah, that's a great question. So one of the other glial cell types that I didn't talk about are oligodendrocytes. Those are for individuals who might not be familiar, those are cells that wrap the axons of neurons with this insulating sheath called myelin. And that's also really important for circuit function, for information to be able to travel rapidly across brain regions. We have not looked at microglia interactions or effects on oligodendrocytes in my lab, but there are other researchers in the field who have been looking at that, and they have seen that microglia can affect how the oligodendrocytes are maturing, and then they can also affect that myelin insulation, how healthy and stable that stays, or whether that starts to break down in different conditions. There are definitely microglia can certainly influence the stability of that insulating layer. So it's a very interesting area.
And then I'm going to end on this question. You mentioned diet, environment. Look, at the end of the day, we want to try to figure out how can we, right, protect ourselves. So what do you have a hypothesis on how you could perhaps a human being alter microglia in a positive way?
Yeah, I think that so in general, like the things that we've all been hearing about that we can modify that are in our control, like having good sleep, having a healthy diet, trying to get some physical exercise, all of those things absolutely influence microglia in a positive way. But I think what one of the things that, and so I would certainly recommend doing that. But one of the things that we're interested in digging into further is some of those lifestyle changes are not possible for everyone. So there are instances where physical exercise is not really possible, depending on if a person has, I don't know, depending on their situation. Physical exercise may not be an option. Um, you may not be able to really achieve healthy sleep patterns if you have a particular job that requires you to have like shift work or periodically work night shifts. Um, and some maintaining healthy diet can also be challenging depending on the circumstances. So we would also like to try to understand if we can figure out exactly how, like, let's say, exercise has a positive benefit on the way that microglia interact with neurons, can we figure out exactly what's happening at that communication between the microglia and the neuron? So we might be able to mimic that or enhance that for people who can't do the lifestyle modification to achieve those benefits. So I think that's work that's still in progress, but in the meantime, yeah, sure. If you are able to improve your sleep and get some exercise and eat healthy, that's always a good idea.
All right. Well, we're going to look forward to some more details on the mimicking
Yes.
of that. So, but thank you so much. I think that this was a wonderful talk. Thank you so much for taking the time to chat about BRF and your Sea Grant and the research that you continue to perform. And I want to thank everyone for joining us today. Be on the lookout for other lunchtime lecture announcements, and we hope you will join us again to learn more about the interesting neuroscience research Brain Research Foundation supports. Thank you all.
Thank you, everyone.